So we've talked a lot about in this chapter brain damage and neurological disorders. In the final section here, we talk about plasticity, particularly in terms of recovery and rehabilitation in response to damage. The first question is at the individual neuron level. So, what is likely to happen if a neuron is damaged? And the easiest way to do this in animal models is to cut the axon of a neuron and study what happens in response. There's three general responses. The first response is that the neuron degrades. Um, the axon degenerates away and toward the cell body. So you cut the axon and it degrades in both directions at different rates, actually. And then once it degrades back to the cell body, the neuron might die. Okay, so you get neuron death. Sometimes that then spreads because we've lost a connection. And because the dying neuron might cause damage to other neurons that are connected to it or nearby. So that's one response. Brain damage can lead to neural degeneration and death and spreading. It's also possible that the axon regenerates, neural regeneration. This is really common in invertebrates. So invertebrates will regenerate neurons pretty regularly in the peripheral nervous system and even in their central nervous system to some extent. This is how you can cut off a limb and it can regrow, and not just regrow the muscles and skin, but um, also the neurons, and they'll come back mostly as normal. In mammals like us, this is a lot more common in the peripheral nervous system than it is in the central nervous system. So neural regeneration is uncommon in the central nervous system, which is why spinal cord damage and brain damage is a problem because those neurons tend not to regenerate. But in the peripheral nervous system, if you get nerve damage in your hand, for example, um, it is much more common that those neurons will regenerate and try to reach back to your skin or your muscles. This is facilitated by Schwann cells, which are glia in the peripheral nervous system. These are the same glia that form the myelin sheath on those neurons. In damage, those Schwann cells that form the myelin sheath are going to help to regrow in the directions it's supposed to go. The third response is neural reorganization, where you don't get that axon back, but the other neurons are going to reorganize themselves. We'll talk about that in a minute. Figure 10.16 from our textbook breaks down axon regeneration and how it's an imperfect process. Okay, so sometimes when a neuron is damaged, when a nerve is damaged, they can grow right back as they were. Okay? And this is typically when a Schwann cell, the cells that form the myelin sheath, is itself not damaged. So if you crush a nerve and you get some nerve pain, but the Schwann cells are fine, then the Schwann cells can allow growth back as normal. Sometimes you'll get imperfect regeneration, and this is typically when maybe the Schwann cells are slightly separated, but aren't completely damaged, and so the Schwann cells can facilitate regeneration, but it might be imperfect, where now this yellow neuron's going to blue, and blue's going to green, and green's going to yellow. And so in this case, you might get feeling back in your fingers or movement control back in your fingers after recovery, but it might never be the same, or it might not be the same for a while, at least, because you've regrown, but it's not quite mapped in the same way. And finally, sometimes axons can't regrow at all, even in the peripheral nervous system. And this is typically when the ends of those Schwann cells are damaged and widely separated from one another, so they can't reach back together to form that connection. Okay, so really is all about these glia in your peripheral nervous system that allow us to regrow. And if the glia are intact, we can regrow well. And if the glia are very damaged, then we may not. As I mentioned, one option for your neurons is to degrade, one is to regenerate, and then the third is to reorganize, right? And neural reorganization is a nice example of plasticity, where your brain actually changes its organization in response to damage. So if you can't regenerate and you can't create new neurons, your neurons can reorganize their connections. There's a few different ways that our neurons can reorganize. The first is dendritic branching, okay, where one neuron leaves and another neuron grows new dendrites to form connections. Another type of reorganization is shown in this figure over here called collateral sprouting, where when neuron A is damaged and degenerates, neuron B forms collateral sprouts to make a connection to the neuron that used to be served by neuron A. And then finally, some of this reorganization can occur not just neuron to neuron, but can be sufficient enough where actual whole areas of your cerebral cortex can reorganize as the um, dendrites and um, collateral sprouts become sufficient enough to actually reach into other parts of the brain. Here's a bit of evidence of cortical reorganization from your textbook. Here you have the normal organization of the motor cortex of a rat, where this part of the brain controls its hind limb, this part of the brain controls its front paw, right? Back paw, front paw, mouth and neck, eyelids. And this red area here controls the movement of its vibrissae, which um, are its whiskers. So the rat can move its whiskers around, and it's controlled by this area of the brain. Well, researchers conducted transection, which is removing this area, removing the vibrissae themselves. Okay? Or removing actually the connection between the vibrissae. You can see that the um, the whiskers are still here, but they removed the connection to the brain. And over time, what you see is that this area of the brain that used to control the whiskers no longer does, and now is involved in controlling the areas of the body that are nearby. So mouth and neck, forelimb, eye and eyelid. So now if you stimulate this part of the rat's brain, instead of moving its whiskers, it's going to move its forelimb. Okay? So this is caused by those changes, collateral sprouting, dendritic branching, actually going to cause a change in the cortex of the rat. Okay. The next pages are going to include a video that breaks down some of these changes, reiterating what we talked about here and animating it. So go ahead and watch that if you want a refresher. Otherwise, I'll see you on the other side.
Some neuroplastic processes help with recovery of function following brain damage. These processes include collateral sprouting, dendritic branching, neurogenesis, and cortical reorganization. In response to a reduction in synaptic communication, axons sometimes develop collateral sprouts. Axon branches grow out of existing healthy axons and form synapses with postsynaptic targets that previously received inputs from now damaged or lost neurons. Dendrites can also change in response to neuronal damage. Brian Kolb and colleagues, 1997, demonstrated that increases in growth of dendritic branching, also known as dendritic arborization, and increased density of dendritic spines correlates with improved motor function following stroke-induced damage in rats. Scientists engaged in laboratory animal research and stem cell research are seeking evidence for adult neurogenesis, meaning the growth of new neurons in response to brain damage. Stem cells are undifferentiated cells whose growth and development is influenced by particular brain regions into which they are implanted. Shear and colleagues, 2004, have successfully transplanted nerve progenitor cells into the brains of mice with traumatic brain injury to the hippocampus. Up to 1 year after the stem cells were transplanted, the transplanted cells had survived, migrated to the injury site, and the mice showed significant spatial learning recovery. Successful stem cell transplantation and recovery of function in animal studies will hopefully lead to similar techniques being used in humans. Ideally, stem cell transplants would replace damaged cells and result in full recovery of function lost to brain injury. Changes in cortical reorganization following brain damage have most often been studied in motor and somatosensory systems. The cortex dynamically responds to changes in inputs. For example, normally, a large area of somatosensory cortex responds to sensations from the contralateral hand and arm. Following brain damage, cortical areas that no longer receive input from a damaged motor or somatosensory region begin to respond to input from other body areas. Ramachandran and Blakesley, 1998, demonstrated that phantom limb sensation and pain are a result of cortical reorganization. Ramachandran's patient, Tom, reported feeling sensation in his phantom arm when his face was touched. The area of somatosensory cortex that responded to input from Tom's hand prior to amputation now responded to somatosensory input on Tom's face. As we have seen, the human brain is a complex and dynamic organ that has the capability, through neuroplastic processes and reorganization, to sometimes recover lost functionality due to brain injury. Though neuroscientists have made great advances in understanding how the brain begins to heal and reorganize after injury, much about the brain's true potential remains a mystery. Results from stem cell and genetics research have offered a hopeful outlook for those who suffer from brain damage. But there is a long way to go before doctors can use these techniques with patients to aid in recovery of lost function.
One of the factors that can help with plasticity or recovery after brain damage is stimulation. And one of the strategies for stimulating the brain is to simply do it directly. um, Plastic changes like reorganization and regeneration in the peripheral nervous system are enhanced when you engage the nervous structures that otherwise wouldn't be engaged after damage. So you can do this directly through transcranial magnetic stimulation, where you pulse magnets on the cortex to stimulate it. So if you have damage to your language centers over here, you can pulse with a TMS pulse to activate those areas to keep them active. Or you can do deep brain stimulation, where you drop electrodes into the brain to activate deeper structures. You can do this for Parkinson's disease, for example, to drop down into the motor coordination areas of the um, of the of the brain, the basal ganglia, for example. You can also do indirect stimulation, so instead of directly um, impulsing a magnet or an electrode, behavioral rehabilitation oftentimes stimulates brain activity simply by having you behave in certain ways. And the general principle, though there's a lot of research done on this, and it's a huge field that you can learn a lot on. But one of the general principles around behavioral rehabilitation is to provide enriched environments and to provide activities like locomotor therapy um, and mirror therapy, which we'll talk about, um, in an effort to stimulate neural reorganization. So if you want to simulate reorganization of motor control along the spinal cord, well, it helps to um, engage in movement of your legs, right? And so locomotor therapy engages movement of the legs so that you can get stimulation through sensory signals, through coordination along parts of the spinal cord that are intact, right? And that activity, just like deep brain stimulation, right, can facilitate plastic changes, cortical, um, collateral sprouting, dendritic branching, that sort of thing, regeneration in certain areas. Mirror therapy, as I mentioned in the last page, is an example of this kind of rehabilitation technique, and this is used for phantom limb syndrome. The video that you watched a couple slides ago talked about phantom limb syndrome being a case of cortical reorganization, where if I lose part of my hand, the sensory signals from my face are going to reorganize and take over activity here, so that now getting a sensory signal on my face might cause a strong oversensitive stimulation of this area of my body, and I'll interpret it as pain in my missing hand. Mirror therapy can cause stimulation of this area of the brain because we got connections between our visual cortex and our motor cortex, right? That dorsal pathway. So looking at my hand in the mirror, even though it's not there, can communicate to my brain that my missing hand is still there. And if I move my existing hand, I may see in my visual cortex movement of my missing hand, then stimulating movement centers and sensory centers of that hand, um, which can then help keep it activated, reduce pain sensations, and coordinate that reorganization in a way that's healthy. So that's it for chapter 10. We talked about brain damage from outside sources. We talked about some neurological disorders, and we talked about neuroplasticity as a general principle and how it can change and recover over time.